about that. Our next speaker is Meenakshi Vengarai at Case Western University, and evidence for lack of bilateral transfer of olfactory learned information in Apis dorsata and Apis mellifera. Over to you, Meenakshi.
Thank you, Clement. So I'm just going to, I think I've skipped ahead a couple of slides. Let me just reshare. Good morning, everyone. And thank you so much to the organizers of the Bee conference for not just giving me this opportunity to speak, but also all of us this opportunity to come and talk about our shared experiences with these wonderful creatures. I'm Meenakshi Vengarai, and I'm presently a postdoctoral scholar at the Case Western Reserve University. I completed my PhD from the University of Hyderabad. And what I'm going to be doing today is sort of describing a small snippet of the work that I did at the University of Hyderabad during my PhD. And hopefully I would be able to showcase to all of you the incredible computing potentials that these honeybees have, with a lot of open-ended questions. So it should make for a fun discussion. I'm sure a lot of you in the audience are aware of this this very interesting tissue that we have in our brains called the corpus callosum. It has been postulated, and I would highlight at this point postulated, that its main job is to shunt information between the two brain lobes. It's a neuron-dense neuropile, and one cannot possibly ignore it. However, it might interest a lot of you to know that the corpus callosum is actually an evolutionary jump. And it made its appearance in the eutherian animals, which are the placental animals. If you go down the evolutionary tree, and if you look at the lower mammals as well, you would see that the corpus callosum does not exist. There are a number of commissures, neurons which go bilaterally, apparently linking information between the two brain lobes, but nothing as visible or evident as the corpus callosum. And then you go further down and you see that insects have bilateral brains. There are a lot of bilateral neurons, especially at the level of vision and locomotion, which help aid coordination. But what about this very important sensory perception that is key to survival for a lot of insects, especially honeybees? What about the olfactory circuit? Do we have bilateral neurons at the olfactory level? So when we look at the olfactory circuit in honeybees, I'm sure some of you are aware, but for those who aren't, the antenna is where the olfactory sensation begins. The sensilla in the antenna will first pick up the stimulus of olfaction or the volatile chemicals in the air. This information is relayed via the olfactory sensory neurons in a hierarchical fashion to the antennal lobes. In the antennal lobes, there is some amount of processing that happens, and there are pathways and neuronal tracts which now relay this packaged information to the higher sensory organs, such as the multisensory integration unit, the mushroom body calyx, which is denoted as MCA. And from the mushroom body calyx, there is a lot of computing that happens. There is a lot of decoding, a lot of packaging. And all this information is now relayed to the higher order learning and memory centers in the pedunculus, such as the alpha and the beta lobes. All this is great. But something very interesting about the olfactory circuit is, so far, especially in honeybees, as far as we know, if information goes up one side of the honeybee's brain, so supposing information is going up the left antenna, it kind of remains in the left lobe, and so is the case for the right antenna and the right lobe as well. So another reason or another, one of the variables that gave impetus to our question was that 2 decades ago, there was this behavioral assay and a set of experiments that were published, which said that if you were to separate the two sides of the bee's head, keeping each antenna on opposite sides of a plastic wall, and you train one side of the bee, over a period of time, 3 hours, there is a temporal shift of this information. And now whatever information is taught on one side is going to be retrieved from the other antenna. And this was very exciting because this is exactly what we were looking for. Not just that, the group also suggested that the animal is able to discriminate between a learned odor and a novel odor. We said that is great. That is exactly what we should be anticipating and expecting. Further impetus was given since my senior in the lab, Shilpi Singh Singh was working on an Indian grasshopper called Hieroglyphus banyan, and using intracellular recordings and electrophysiology, she was able to locate a bunch of neurons in the grasshopper, which were located ventrally in the mushroom body calyx, and they had processes which integrated across the two sides of the pedunculus, and more importantly, the alpha lobes, which, like I previously mentioned, are considered to be higher order learning and memory centers. Further, she also saw that these neurons that she was able to fill with intracellular dyes were weakly responsive to her panel of testing odors. So we jumped straight in. We said, okay, we have some kind of background. It looks like perhaps bees might be able to have this computing potential where in some way information from one side is getting shifted to the other side. I was very fortunate to work with two species of honeybees. One was Apis dorsata, the other, the Western honeybee Apis mellifera. Apis dorsata is a wild species of honeybee. They grow on tall trees, and you cannot domesticate them. My lab senior at that time was in fact characterizing the olfactory system, and she was also able to provide me evidence that both the olfactory systems are nearly identical. So we said, great, we will just go ahead with Apis dorsata, and we will see if wild honeybees perform at the same level as the Western honeybee. It was a three-tiered approach that I used. We used electrophysiology, neuronal tract tracing, and behavior to approach our questions. And just for the want of time, I'm going to quickly skim over some of the work that we did and get to the behavioral results. This is a quick overview of the electrophysiology setup that we use. We have two electrodes. One is the ground electrode, the recording electrode. We place it in the animal's brain, and the signals picked up are amplified in an amplifier, digitized in a digitizer, and you have an oscilloscope also to view the signals. So the first thing that I had to do was, in my experiment, make sure that I have the right block. And just by chance, serendipity almost, we found acrylic paint lying around in the lab. And what we had to do, and bear with me while I quickly explain what it is that we did in our paradigm. The first thing that I did is make sure that this block does not harm the antenna when I shut it off, and it is reversible. That is, when I coat it and I remove it, the antenna is still able to respond to stimulus, which is what you see in this recording of an electroantenogram, where the blue line is before coating, the red line shuts off the odor stimulus, and the green line shows that the response has been restored. The next thing that we did was, if at the physiological level there is any bilateral transfer, if I shut off the ipsilateral antenna and I stimulate the contralateral side, technically the mushroom body calyx on the shut side should start responding to this stimulus. However, we see that at the physiological level, when I shut off the stimulus on the ipsilateral side, the mushroom body calyx does not receive any signal from the contralateral side. We proceeded to also check for the same kind of possible contralateral connection at the alpha body level, where we carried out simultaneous recordings from both the alpha bodies. I will get back to this result if somebody would like to perhaps discuss it further. But the next, what I can definitely tell you is we were not able to see any contralateral connections at the physiological level. So we said maybe at the anatomical level, we'll find something. And we carried out neuronal tract tracing. And even for this experiment, what we did was we used dyes, two differentially colored dyes, and we tagged anatomically relevant regions on the opposite sides of the brain. So for example, I'm showing you one set of results that we did, one of the few sets of results that we carried out, where we tagged the mushroom body calyx on one side, and then we tagged the alpha lobe on the other side. And what I would have anticipated, had there been any contralateral connections, would be that there would be some kind of a pathway created between these two areas. But as it turns out, both of them meet dead end in the center. Very interesting. And still, as our hopes dwindled, we hope we would find something. Unfortunately, I was not able to see anything even between the antennal lobes in the mushroom and the alpha lobe. And we made many such pairings and combinations. So we said, but hey, they saw something at the behavioral level. So maybe we should be able to see something at the behavioral level. And we designed our behavioral assay. So the behavioral assay that we designed required us to carry out Pavlovian conditioning, where the insect, or in our case, the honeybee, is provided a response to stimulus. and that is overlapped with a reward. In this case, it is a sucrose reward that we provide the honeybee. And the honeybee, over a period of many trials, provided there is a clean intertrial interval, the animal will extend its proboscis in anticipation of the sugar reward. And this was the basis of our behavioral assay. To check for the bilateral transfer, we had two experimental groups. The first experimental group, we would block one antenna, train the animal with the open antenna. Over a period of 3 hours, we would now shift the block to the trained side and check from the untrained side. If there is any kind of bilateral transfer, technically the animal should start responding now when I provide the same trained odor to the untrained side. We also kept one set of experimental animals just to make sure that there is no loss of memory in general. And in this case, our animals had one block on either antenna, and that was maintained for 3 hours. We checked for retention from the same trained side. I would also like to highlight at this point that the experiment started with Apis dorsata, but given the results that we were seeing, we also ended up working with Apis mellifera, and the results were comparative. So panel A is Apis dorsata, panel B is Apis mellifera. Panel A, I would draw your attention to the dashed line, n is equal to 51, where we see that the animal with one antenna is able to learn information up to the 5th trial. Clement, am I out of time? I'm sorry, I think you're on mute.
3 more minutes.
All right, okay. So the animal is able to retain information. However, when I check for retention from the untrained side, I see that the information retention drops to close to 0. The same results were seen in Apis mellifera, and Apis mellifera has a much more robust learning and retention rate compared to Apis dorsata. The second set of experiments was to make sure that the animal is not generally forgetting. Apis dorsata does not, in fact, forget the learned information. There is a 3-hour retention rate similar to the learning rate. With Apis mellifera, we decided to take it up a notch, and we see that the same set of animals that were not able to retain information from the untrained side were able to maintain the information in the trained side at 3 hours, and there is also a good amount of discrimination. But what is it that we were missing? So we decided to recreate the experiment which set all of this off. And we see that when we use a plastic wall to separate the two sides, as described by the group, I changed this variable a little bit. I trained the animal with one side blocked with my acrylic paint, now that I knew that acrylic paint was a good block. And I see that over a period of 3 hours, despite the other side not being exposed to this odor, the animal continues to learn, which most probably reflected that there should be some kind of leak in the system. It does not change the fact that of course, we carried out a set of experiments which showed that the block was not acting as a contextual stimulus. Just to wind up, we still anticipate that the reward pathway, because one cannot deny that there is at some level the tying up of information that is happening. And what we anticipate is it's very possible that honeybees are tying all this information that is coming in at the reward pathway, because that is something that plays a very vital role in their decision making as well. I would be happy to go over this if anybody would like to discuss after this meeting. But just a quick conclusion. We do not see any direct bilateral connections at the olfactory level, both in Apis dorsata and mellifera. Another interesting conclusion that we were able to draw was that the probability of evoking a proboscis extension reflex, especially in Apis dorsata, was very predictable. And we are presently looking for reward pathways in the lab. My junior is taking on this project, and we're very excited because we think we might be onto a model for split brain studies. With that, it was a very big collaboratory work in the University of Hyderabad. This is the teams. Thank you so much. I'm open to all the questions.
Thanks. Wonderful work. And I say this having worked in fly labs where people try to do similar work, so I know how much work it is. Benjamin says that it's a wonderful talk too. How do you think the learning might differ if you used PER as the learning assay? I thought you did.
That's what I did, yeah.
Yeah. Did you notice?
PERs. Yeah.
Did you notice any differences across caste or sex?
Oh, so in terms of as in foragers and nurses, is that the question?
I guess he's asking about, did you do queens versus workers or
Oh,
drones
no. We
versus-
were looking solely at foragers because it started off as we wanted to look at mostly olfactory as a computational variable. So we were more keen on, keeping the foragers as our model of interest. So we looked at only foragers, but yes, it would be interesting to look at the queens. That would be great. Thank you.
Okay, thank you very much.